Good afternoon and welcome, honored speakers and guests, to the 2021 John R. Freeman Lecture. Today's event is being recorded. I'd like to say just a few words before we move to the main program. My name is Karen Kelly, and I am the chair of the Freeman Committee. The committee is part of the Boston Society of Civil Engineers section of the American Society of Civil Engineers, which sponsors this annual event. Our event today is the latest in a series of distinguished lectures held since 1966 in honor of John Freeman. Mr. Freeman was a graduate of the civil engineering program at MIT, and he went on to become one of the most renowned engineers in America in the early 1900s. Locally, he was responsible for the design of the original Charles River Dam, and nationally, he helped design the water supply systems for a number of municipalities, including San Francisco and New York City. Today's event derives from a gift Mr. Freeman made to BSCES in 1925, nearly 100 years ago, expressing his wish to inspire young people to take up a career in hydraulic and water resources engineering. In that spirit, and on behalf of the entire Freeman Committee, we hope this annual lecture will help convey the excitement of the civil and environmental engineering profession. And we hope that if you are currently pursuing studies in civil engineering, that like Mr. Freeman, you will find a home with BSCES. With that, I would like to introduce Bruce Jacobs, who will say a few more words about BSCES and our sponsors.
Very good. Thank you, Karen. So my name is Bruce Jacobs. I am president of the Boston Society of Civil Engineers. And I particularly want to speak to the younger members of our audience and just kind of relate to you my own experience with BSCS. I first became involved actually by attending one of these Freeman lecture events. And the president at the time did essentially what I'm doing now, put a bug in my ear and said, I should really get involved. I became involved with the Environmental Water Resources Institute, otherwise known as MWRA. I was already probably pretty late in my career from your own perspective, but became involved through that later, began involved in more kind of leadership positions. And I will just say it's been highly satisfying, the number of people you meet outside of your own company, clients, the professional advancement and the technical talks that I've attended are just amazing, and you're just not going to find it anywhere else. And the networking opportunities are just enormous. So I highly encourage you not to wait till you're late in your career, as I did, and get involved now. There are tons of technical groups and other groups you can get involved in. And if you don't know where to start, I'll just recommend that you email me. And I have an easy email address to remember, president@bsces.org. And I'll work with you and we'll figure out like where your interests lie and what group might be best fit for you. Okay, so please do that by all means. I have to do something that, which is also thank our many sponsors that help pay for the event that we're attending and other events. And the society sponsors are AECOM, Earthsoft, GI Consultants, and Keller. And our program sponsors are Alfred Benesh, Beta Group, BSC Group, CDM Smith, Childs Engineering, CN Bro, Collins Engineering, Dewberry, Green International Affiliates, GZA, Haley and Aldrich, HMTB, Horsley Witten Group, Howard Stein Hudson, Hoyle Tanner Associates, Jacobs, JS Held, Kleinfelder and Niche Engineering, Northeastern University, Sanborn Head and Associates, Simpson, Gumpertz and Hager, Skanska, Stantec, Ty and Bond, Transystem, Trebico, Tufts Engineering, Weston, Sampson, and WSP. If you haven't heard your name, your company's name was up here, you should find out why and figure out how to become a sponsor. And I want to have one more minute, and it won't take very long. Just so you know, for a lot of people, you probably recognize the Orange Journal. We're publishing that again. It's online only. So if you have any interest in publishing, particularly about like local engineering things going on, I encourage you to do that. Also, you can serve on the editorial board. I also want to let you know tomorrow evening is our awards event, and we are honoring Jim Lambrechts from Wentworth University, formerly the editor of the journal, and from Haley and Aldrich. Please do attend. You can find out more information how to register on our webpage. And lastly, and this is the last thing I have to say, is we have next week a fantastic event coming up on offshore wind. And we're going to have several speakers from Vineyard Wind coming in to speak. It's on June 22nd. Again, check our BSCS webpage to get more information. So that's all. Thank you very much. And again, email me if you have any, if you want to get back to me.
Thank you, Bruce.
Very welcome.
I'm Steve Smarjassi. I'm Director of Planning, and it's my privilege today to introduce our panel. Along with all the pain and suffering and loss that the worldwide COVID pandemic has caused, it's also sparked new interest in using wastewater as a means for population-level disease tracking. Advances in RNA detection now allow wastewater to be used as a snapshot of the health of a region, a community, or possibly even an individual facility. Massachusetts has been a real hotspot for the development of these techniques. For today's Freeman lecture, we've asked a distinguished panel of experts currently using those techniques to discuss the state of the science, how it's helping manage the pandemic, and what the future of these techniques might hold. I'll introduce all four of our speakers right now, and then let each of them provide a brief perspective on how wastewater surveillance works and is being used. Once they've all spoken, we're going to open it up for questions. Feel free to put your questions in the Q&A at the bottom of your screen, or perhaps at the top of your screen at any time, because we'll be using that during the session. Dr. Monina Klevens is Director of Research and Evaluation in the Bureau of Infectious Disease and Laboratory Sciences at Mass Department of Public Health. She's been coordinating their surveillance of wastewater for SARS-CoV-2. Her 30-year public health career in infectious disease epidemiology started in CDC's Epidemic Intelligence Service program and has included extensive work in immunization, HIV/AIDS, viral hepatitis, healthcare-associated infections. She's published and lectured in the US and abroad, is a member of the Society for Healthcare Epidemiology of America, and a fellow of the Infectious Diseases Society of America. Dr. Claire Duvallet is a senior data scientist at Biobot Analytics. She coordinates a team of researchers spanning computational biology, environmental engineering, applied statistics, and epidemiology to derive useful public health insights from Biobot's wastewater dataset, to seamlessly connect Biobot's data science across their data science, software engineering teams, and to communicate the science behind their data and its public health implications. Claire received her PhD from the Biological Engineering Department at MIT, where she studied the relationship between the human biome and health and disease. Steve Rhode is Director of Laboratory Sciences at the Massachusetts Water Resources Authority. He has over 40 years of experience in environmental chemistry and microbiology, including 27 at the MWRA. He directs the operation of 5 MWRA laboratories that support MWRA operations from Deer Island all the way out to the Quabbin Reservoir. Steve has been MWRA's lead technical person on COVID science throughout the pandemic. Dr. Anna Mehrotra is technical leader at CDM Smith's wastewater process engineering practice. With more than 15 years of experience, she's an expert in industrial and municipal wastewater treatment design, including process modeling, specialized sampling, and pilot studies. She's a licensed PE with a master's from Stanford and a PhD in civil environmental engineering from UC Berkeley. She's currently spearheading CDM Smith's wastewater surveillance program, overseeing efforts in Egypt and Jordan, as well as across the US from California to Massachusetts. And with that, I'll hand it over to Monina. Looking forward to this exciting presentation.
Thank you very much. It's an honor to talk to you. And as a Boston resident, I also wanted to say thank you for all the work that you do in infrastructure and even more in preparing for adapting and for climate change. So the Bureau of Infectious Disease and Laboratory Sciences, pictured here in the lower right, is located in Jamaica Plain, and it is on the corner of the Arnold Arboretum, and there are offices and the state lab is located there. Next slide, please. I wanted to say that we actually haven't contributed a lot of the original science, but actually are building on what is already a public health infrastructure. In 2003, it was used, this is a document on your left, from the WHO to monitor poliovirus in Africa and other areas. And then there was a summit, a global summit, before Massachusetts started to use this technique for monitoring SARS-CoV-2, and that really provided a lot of the support for us to gain some already lessons learned before we embarked on this in Massachusetts. Next slide, please. The strengths of wastewater surveillance for public health are several. Number one is that the sample is unbiased, which is a difficult place for us to find information such as this, where these provide information on a pooled community sample. It's confidential, not traceable back to an individual. It's not affected by certain limitations of clinical indicators and of the behaviors of the individual. For example, sometimes our diseases depend on an individual coming to seek care before it can be identified for surveillance. And in this case, the infection frequently was asymptomatic. Those viruses also are in the wastewater that surveillance wouldn't know of otherwise. The wastewater allows us data for communities where timely testing might be challenging, and I'll show you an example that we're using in that use case. It's also available at a smaller level than the sewer shed. It could be county, for example, or it could be depending on the distribution of the area. It could be a whole campus. It could be a specific neighborhood or a building. And I put may precede because as clinical testing became more timely, it's unclear, and this is a place where I think we need to continue to monitor how well wastewater precedes infections in the community. Next slide, please. This I wanted to show you on the left is that public health depends on many sources of data to be able to put together a composite of the full picture. We have sources of data that give us information on all of these levels. The wastewater component gives us information at this broader bottom of the community level, because otherwise, as I mentioned earlier, if people have to come and seek care, we can get information captured from ambulatory care. We can capture information on hospitalizations in the most severe, and in this infection, also the cases who had a death as an outcome. We can monitor all of those pieces. So back to the community infections, what wastewater contributes to public health is in the area of a general warning, because as I mentioned, it doesn't give us the information that is about the individual. It's community trends that we're looking for. As vaccination was implemented and other mitigation, non-pharmaceutical mitigation strategies were implemented, the wastewater trends also allow us to monitor the impact of those. And most recently, we've been able to use it to detect the appearance of variants of concern. It helps identify for public health communication when you can tell a community that this has been detected, that it reinforces the message for prevention and mitigation, and helps us to protect the community through this outreach so that they can be more vigilant and protect themselves. Next slide, please. And this is exactly what the program consists of in Massachusetts currently. And that is, we have 2 contractors, Biobot and CDM Smith, who send information to the health department. In turn, we have several different programs. We're using the data from MWRA, and I'll show you, and then you'll hear more from Steve Rhode. But also we had at the city level, 5 different locations that has now backed into 2 different locations to have broader coverage. We also have a use case in the Department of Corrections where the Department of Corrections identified 9 priority sites, and I'll show you some of those results. And finally, in 2 of the soldiers' homes, where this has been a supplement to clinical information. This is what I wanted to show you about how we're using the Department of Corrections monitoring so that we can compare them to each other using the normalized values that come in the report that we receive from the lab. And putting this together, you'll see, or, maybe because it's small, you can't see so well. Thank you for that arrow. The MWRA Northern and Southern are here for a comparison. And without this normalized value, we wouldn't be able to compare it all. So this is helpful for context. Next slide, please. There was a question about whether communication of the results was a missed opportunity, and I hope we'll hear more about this, in that from the state perspective, we used this as one of the tools in our armamentarium, but didn't really maximize communication of this to the public. It's unclear what the best strategy in communicating that is, because communicating risk in general is difficult. So instead, what we did was to limit the feedback of the results to the partners. So for example, in the scenario that it was the city of the locations feeding that information directly to them, to the Department of Corrections also, and the soldiers' homes to the facilities and the management there. But there was a missed opportunity potentially where, for example, this is just one example, Rachel Maddow, who has wide visibility, asked for interviews, and we didn't take her up on that. So potentially a missed opportunity for communication. This is actually a project that we do have that we hope to contribute in the area of the science. The question is wastewater surveillance a cost-effective option for measuring COVID-19 cases at the correctional facilities in Massachusetts? And this is the example I referred to earlier about where testing can be challenging. And here, this is work that in our office, Lori Chow is conducting, and it is superimposing this model of a decision analysis on a standard infectious disease dynamic SEIR model, and that is work in progress. Next slide, please. Of course, epidemiology accepts that everything has limitations, and wastewater in public health has limitations too. In the US, this is a CDC slide. In the US, about 25% of residents are not connected to sewers. In Massachusetts, it's more like 30%. These sometimes decentralized wastewater treatment facilities will not be captured unless you went to some system of a sample, for example, of septic systems or something along those lines. Low incidence may be below the limit of detection. Lab assays have limits in sensitivity and specificity, and that we should avoid the use of interpreting low or negative results as a mechanism to say that a community is clear. This is where it comes into that pyramid where we need to put several sources of information together. And then finally, it may be impacted by pre-treatment of sewer waters for whether it's for safety or for odors, where, for example, in the Department of Corrections, this can happen where if there's bleach, then it can affect the results. Next slide, please. This is to show you where we hope to go in the future. We want to expand partnerships of who we are working with. We've reached out to the Department of Environmental Protection. We want to also expand coverage from the few areas that I showed you in our fragmented current approach to cover more of Massachusetts, and to expand in the targets. Right now, it's only COVID, SARS-CoV-2 that we're looking for, but we would like to include flu, potentially other coronaviruses. This might be also a place for hepatitis A and other, for sure, antibiotic resistance targets. The other thing we'd like to do is to bring testing in-house so that this is something that will save us funding. And then this is a resource available on the CDC website. They are recruiting states across the US to participate and submit information through this national wastewater surveillance system. And they also have in mind funding for other targets moving forward. So we think that this is a platform that is going to yield fruit for a long time to come. Next slide, please. This is our website, and thank you again for your attention, and I will pass it to the next speaker.
Thanks, Monina, for that great overview. So hi, everyone. My name is Claire Duvallet. I'm a data scientist at Biobot Analytics. And today I'm going to tell you a little bit about our company and our work in general, and then specifically with COVID. So for those of you who are new to this field of wastewater epidemiology, you know, the concept is really simple. When you use the toilet, you're essentially flushing a high quality medical sample down the drain, and that sample is then collected in sewers where it's aggregated with other individual samples. And so much like, you know, your doctor looks at your stool or your urine sample to understand something about your health, we at Biobot view wastewater as kind of a community level stool and urine sample. And so it can tell you something about population health. So Biobot kind of started as a, out of an academic collaboration at MIT, kind of propelled by this idea of sewage as a source of population health data. We were started about 4 years ago at the time when wastewater epidemiology was still really, really niche in the US. And at the time, the largest public health crisis in American history was the opioid epidemic. So that's where we started. And we started by monitoring metabolites that are excreted after consumption of licit and illicit opioids, as well as treatment compounds and other chemicals of interest. In the next slide, I'll show you a little overview of some of our initial work. So we first started with a pilot study in North Carolina, funded by the Bloomberg Mayor's Challenge. This 6-month pilot had really positive results and actually converted to a full contract. So, you know, like a line item in the city budget, which was very exciting for Biobot. And kind of 2 stories that came out of that work. So first, we heard from the town of Cary that they really appreciated having this partnership and having this data in their community to go out and have conversations with citizens about the opioid epidemic, where previously, you know, it was kind of viewed as not an issue there. And using our data, they were able to go out and show that the opioid epidemic was a problem kind of everywhere and help reduce stigma and do outreach that way. They also used our data to kind of inform where they put their booths for National Prescription Drug Take Back Day. And because of that, they kind of moved the booths to the suburbs. And that year they saw over a 2-fold increase in the amount of medications disposed of safely. So, you know, this was really exciting work. We were ramping up and starting to work across different cities in Massachusetts with the Department of Public Health and getting ready to scale up across the US. That was in early 2020. So that was, you know, a little bit interrupted by COVID. So essentially, as soon as the first reports of the SARS-CoV-2 virus being shed in stool was reported, and I think around January or February, we were hard at work, you know, developing and adapting methods to do this monitoring regularly in wastewater. As a public health company, we knew we had to respond, and so we were kind of all hands on deck. So we collaborated with the Alm Lab at MIT to kind of rapidly develop the methods to detect the SARS-CoV-2 virus in wastewater. And this all happened really rapidly. You can see in the graph, these are samples from Deer Island that came from mid-March. We launched a nationwide pro bono campaign to scale this technology across the US on March 25th. We initially had room, or we had room for 100 locations, and we were like, okay, if we hit 100 sampling locations, we'll be really excited. And in the next slide, you can kind of see the scale of the pro bono campaign. We hit 100 facilities within like the first week or so, had to pause, regroup, get ready to scale up, and then reopened sign up for facilities. And through that campaign, through the summer, ended up covering over 40 states, representing over 10% of the population, which was really exciting and kind of a great way to collaborate with facilities and academics to really develop the methods. We transitioned to a full service offering in June and have since been offering wastewater surveillance as a regular service for hundreds of locations kind of weekly. We've also continued to work to develop the technology. And like Monina mentioned, we were excited to launch a variant detection add-on in April. And then recently have actually gotten a federal contract to expand nationally. So the map in the previous slide is now updated, and we're actually operating in all 50 states, which is really exciting. From the perspective of the sampling locations, which I think Steve will dig into a lot more, how it works is that participants sign up and order their kits online. We then send them a shipping kit that has everything they need to collect a sample, really trying to make it easy and trying to make it fit within existing sampling operations from these facilities. They mail us back raw sewage to our lab, which is in Massachusetts, in Cambridge. And then that's where we do our chemistry and molecular biology assays to measure SARS-CoV-2 and a variety of other targets. The data then goes to our computational biology and data science teams, where we do our very best to interpret it and derive useful insights for folks like Monina, for the public health folks on the other side. And then finally, we package all of the data in visualizations and a report that we send to our sampling locations and their partners. And, you know, it's been a really exciting year for Biobot and also just for the field of wastewater epidemiology in general. I think, like I mentioned, when we started this, no one had heard of the field, and now here we are having, you know, a really exciting panel with a variety of stakeholders talking about this technology. And this is just a screenshot of about 50 different sampling locations where we've been working over the past year. And the blue lines are the wastewater concentration, showing a 3-sample average. And then the orange line is the reported cases in that associated county. And so, you know, the data is really noisy. I think both wastewater data and clinical data have known weaknesses and limitations, and one of those is noise. But it's pretty remarkable if you kind of step back and blur your eyes just how well the data has been tracking. And so, you know, I think it's been really exciting to see how well wastewater is performing and really reflecting those trends in communities. And that gives us a lot of excitement and kind of validation for this technology moving forward. And so, you know, just reflecting a little bit on our role within this entire ecosystem, I think it's kind of encompassed by this idea of, you know, implementing wastewater epidemiology at scale. So we've worked really hard to optimize for operational and logistical constraints. For example, something we heard early on was that getting results really quickly was extremely important to our participants. And so we made sure that our lab methods, you know, were really well suited for rapid turnaround times. We're also making, you know, auxiliary tools to make it easy for participants to do this wastewater sampling, from the sign-up flow to the kits to the reporting. We're also collecting, you know, a really large data set on wastewater SARS-CoV-2 concentration. So we've tested, I think, close to 10,000 samples. And what's important is that means that the insights we get from this data are really generalizable across wastewater treatment plants. So across large and small treatment plants, dry and wet conditions, our data kind of covers it all. And so if you can see patterns in the data we have, we're pretty confident it'll apply kind of no matter where you go. And then finally, we're really excited to play a key role in the whole network and in collaboration between, connecting wastewater officials with public health officials, and then also serving as kind of a contributor to the scientific community so that, we can expand this field as a whole. And more broadly at Biobot, we really think of sewers as population health observatories. So this implementation at scale is actually not just about how many locations we're sampling in, but also the sorts of things we're looking at. And we're actively working to develop wastewater as a platform. Like Monina mentioned, you can measure a variety of other targets in the wastewater, from infectious diseases like Zika, influenza, norovirus, to other chemical markers of, for example, consumption with opioids and other licit and illicit drugs, diet and nutrition, as well as just general markers of health, of chronic disease, of stress. Again, anything that's in your stool or urine, we can measure in wastewater. And through COVID, we've really seen the potential for wastewater epidemiology to become a standard part of the public health toolkit. And it's been super exciting to see folks like Monina and Steve really take this up and implement it into their regular operations. So we're really excited for wastewater to be, a key component of existing public health infrastructure and wastewater infrastructure to combat future pandemics. And from our perspective, wastewater is really useful because it's predictive. You can see the results in wastewater maybe before folks have reached the clinic to get tested. It's inclusive. Everyone has a voice in the sewer as long as your toilet's connected to a sewer system. Your data is going down there and can be measured. And it's versatile. We can, from the same sewage sample, measure a large variety of markers that can tell you about many different things about population health. So that's a little bit of an overview of who we are, what we do, and why we're excited about this. And now I'll hand it off to Steve to tell you, boots on the ground, what it's been like.
So I'm Steve Rhode, and I am the Director of Laboratory Services at the MWRA. And I'm going to give a view of what it's like as a utility, and also discuss some sampling aspects of it. But since the pandemic started, we have been working with Biobot on sampling wastewater to track the COVID in our north and south systems, and we post the results on our website. And as Monina mentioned, it's gathered some national media attention as well as local attention. The Globe regularly posts our data as one of the key graphs to watch as how the pandemic is proceeding. Next slide, Steve. So the first time I gave a talk like this was a little more than a year ago, and at that time there were only a few places in the world that were publishing results of using wastewater as a predictor of COVID. And courtesy of the University of California at Merced, they maintain a dashboard now of all of the places in the world that are publicly sharing their data, and you can see that it's a lot of places in a lot of countries, over 50 countries and over 2,000 sites around the world that are being posted. In addition to our work with Biobot, we have contributed samples to pretty much anyone who asked. We at MWRA view ourselves, people don't often think of their wastewater agency as a public health agency, but really we are, and we certainly think of ourselves as that. And so we're happy to do anything to help out with any sort of research that will help control the pandemic. Next slide, Steve, please. So just to go through the timeline of the work that we have been doing with Biobot, it actually goes back to 2017, 4 years ago, when Biobot had just formed, and prior to the study that Claire talked about in North Carolina, were doing some preliminary work in Cambridge in Massachusetts, and they came to us, Marina and Mariana and Nusha came to us because they hoped that we had a single sampling site that would allow them to look at the whole city of Cambridge. And unfortunately, our system is not designed that way, and so we weren't able to help them. You know, as Claire mentioned, in March of 2020, they approached us again and said, you know, we think that we can find this virus in wastewater, and can we get some samples? And we were like, sure. And it was literally like 2 days later, my phone rang, and it was Mariana, and she was really excited. It's there, we found it, it worked. And I was like, you know, my first question, because I'm a lab guy, was so what's your negative control? How do you know, what other wastewaters have you tested? And she's like, well, we're still working on that. And I said, you know, we have archive samples. We archive our samples typically for 3 months. So we had samples back to January when, at least at the time, until yesterday, we believed there were no cases in Massachusetts. So we sent some of those. And then, you know, later, Dr. Alm actually came back and asked and said, you know, could we fill in the gaps with a couple samples a week and maybe every sample for early March? And we did, and that was the basis of the paper that Claire mentioned in her talk. And at the same time, they were working with Boston Water and Sewer Commission to look at neighborhood results, and they were hoping to use that data to calibrate cases, you know, in the population to the wastewater numbers, and that data is also discussed, I believe, and I think there might be in the second paper. So our current program is we sample 3 to 7 times a week. We've actually been 7 times a week since the beginning of the fall winter wave, and have been, you know, sampling every day, basically at 2 sites. So, you know, we increased to daily if it looks like there's a lot of activity going on. We share the results immediately with the State Emergency Command Center as soon as we receive them, typically within an hour. Both Claire and Monina already mentioned that Biobot had added the capability to do variant prevalence for what's now called the alpha variant, was previously known as UK or B1117. Those data are also posted. All of our results get posted a couple hours after the command center has had a chance to look at it at MWRA.com. Next slide, please. So one of the questions that we always get back is, you know, is it possible for people to get this from wastewater? And the first and most obvious answer is there are always things in wastewater that you should avoid unprotected contact with it directly, regardless of whether there's a pandemic going on. But the second answer is that you know, SARS-CoV-2 is an enveloped virus, and enveloped viruses don't actually survive very well in water systems or in the environment. And you know, most waterborne diseases are actually non-enveloped viruses because of that. And in order to stay infective, you know, this is a little bit of virology 101. You know, the virus consists of an envelope, a protein capsid, and then the actual RNA or DNA. And what, you know, Biobot and other people around the world are testing for is actually just small pieces of that DNA. And as long as those small pieces remain intact, you can still detect it, even though most of the rest of the virus may have completely degraded away. And so, you know, the CDC has been looking for, you know, the possibility that people could be infected through wastewater. They haven't found any data to that effect at this point. And I can say anecdotally that we at the MWRA, you know, we have staff who have contact with wastewater. We have other people on the drinking water side who do not. And we have a group of people since, you know, March of last year who were teleworking 100% of the time. And the cases of infection that we've seen amongst our staff are really evenly spread among those 3 groups. And so there's no indication that people are getting sick from the virus in the wastewater. Of course, the first thing everybody wants to do is look at the data and say, you know, convert that to numbers of people that are infected. And it's really difficult for a couple different reasons, although Claire has made a lot of progress in kind of getting those numbers better. But in reality, the patients who are shedding, it's over 6 orders of magnitude, from 100 copies per gram of feces to 100 million copies per gram of feces. And so that makes it inherently difficult to put a number, or to translate the number of gene copies that you see in the wastewater to a number of people. And then that's further complicated by just the nature of wastewater itself. It's really a variable system. In a lot of systems there are rainfall effects. Even people who don't have CSO see some rainfall effects, and there are other things. Wastewater is really not a homogeneous sample matrix. Next slide. So here's an example. This is actual flow data for a 2-week period from a couple weeks ago when I was putting my slides together for this. The blue line at the top is the flow data for our north system, which has CSOs. The red line is the flow data for our south system. The green line is actually the south system at the other end of the 5-mile tunnel that feeds it over to Deer Island. There are no CSOs on the south system, and you can see there's a very steady sort of diurnal, but you can see around the middle there, above the 14 days, that large rainfall does still affect that flow some. And you can see there's a clear diurnal signal during dry periods, and then wet signals that get flushed out. One of the interesting things is that suspended solids doesn't really correlate directly with flow. So people usually look at this graph and they say, well, okay, so it gets diluted when the high flow, and you just adjust for the volume of the water and the numbers all come back into line. But the reality is our suspended solids don't correlate directly with that flow data, either directly or inversely. And what we see is that the solids, I think, don't travel at the same pace as the wastewater. And so one of the effects that we can see is that if it's been dry for a while and then we get a rainfall, we will see a spike in the solids concentration, and then for a few days after that we may see a drop. But other times it will track along with the flows, and so it's difficult to predict. Next slide. So here are some implications of how things behave in wastewater and how they sample. The graph on the left is sort of a ubiquitous soluble indicator. The people who put this paper together actually use sucralose, and that will typically just follow a diurnal variation, and because it moves directly with the wastewater and it's being input all the time. I believe sucralose is probably more in the urine than it is in feces, and so it just moves along with the liquid stream. The example in the middle is if there's a rare contaminant, or you can also think of this as one person in a dorm who's infected. And so very close to the dorm, you get a very narrow spike when that person uses the bathroom, and the rest of the time there's no signal. As you move a little bit further downstream, that does disperse some, and it also gets diluted as it moves along, but it does tend to move as a slug through time. So how do you get around that? You need to, the closer you are to what you're trying to sample, you need to sample more frequently or continuously if you can. You can offset it a little bit with flow triggered sampling. So somebody flushes a toilet and the sampler sees that flow and it samples, but of course it's also going to pick up if somebody's in the shower or the dishwasher's draining and stuff. But if you composite it all together, that's still you have a more likelihood of catching that narrow spike than you would if you're just sampling every 15 minutes, which works really well for us at the wastewater treatment plant, but it doesn't work so well at places that are closer to the source of the contamination. Sampling from places where there's some kind of mixing also works. In fact, a number of researchers prefer to sample primary sludge at the treatment plant because there's an inherent, the solids settle out at different rates, and so they're sort of inherently composited over time as they're collected, or sampling downstream further in the facility, or downstream further in the sewer system. Next slide. And actually, this is my last slide. This is the actual data with error bars that were developed by Claire at Biobot. And you can see that it's fairly noisy. So the green dots and the green part of it is our north system. The orange is the south. That's backwards from Ireland. If I had realized it at the time, I might have flipped them, but it is the way it is now. And you should also note, for looking at our data in particular, that Boston, Brookline, and Newton are significantly split from, some of the flow goes south and some of the flow goes north. And so, Boston in particular still tends to drive our numbers for everything that we look at in terms of the water and the wastewater. But you can see clearly that, we had a spring wave, and then the fall to winter wave, winter spike that, has taken some time to go down, and all the way over at the right side, you can see where we are. The numbers have been running very low, which is consistent with the way things went in terms of cases and other indicators that are tracked by public health. And I will stop there and pass off to Anna.
Thanks so much, Steve. And thank you, BSCES, for the opportunity to be here today. So I'm going to close us out and give you the perspective of a consulting engineering firm working in this space by talking about CDM Smith's experience with wastewater surveillance over the past 15 months. So I'm going to answer a series of questions. But if I had to sum up our experience in a single word, that word would be sewers. We've gotten up close with sewers a lot in the past year. So let's dive in, and I'll show you what I mean. Next slide, please. Great. So first question is, how has our work fit into all the wastewater surveillance that's being done really around the globe? Well, one of our most important roles has been as wastewater sample collectors. So we are collecting grab samples every weekday from manholes at 8 VA sites across the country. We are babysitting auto samplers deployed in manholes at at 2 soldiers' homes in Massachusetts that you heard Monina mention. We are filtering wastewater in the field, so right at the manholes in 9 different neighborhoods in southeastern Michigan. We are part of a collaborative surveillance effort in the city of Chicago, where, you guessed it, we are collecting samples from manholes. So really, we are the boots on the ground. Most of our surveillance activities have been small scale, so focused on individual buildings, facilities, portions of neighborhoods and zip codes. So rather than collecting samples at the influent to the wastewater treatment plants, although we do that as well, we are really focused on pulling the samples directly from the sewer so we can get that more granular information we need to track COVID. But we also use our sewer knowledge for our surveillance projects in other ways. So we can be the GIS specialists who define the population in and the geographic extent of a sewer shed that's captured by our sampling manhole in Southfield, Michigan. We can be the hydraulic modelers who can estimate the flow at a Revere, Massachusetts neighborhood sampling location. And I will say we've also dipped our toe into research, having evaluated, how easy is it to get up and running with the Luminultra gene count PCR device. It's a relatively portable, easy to use PCR machine. And the answer is it's pretty easy to get up and running. And we've also compared grab and 24-hour composite samples and found that, well, yeah, they're pretty comparable, but not identical, as you would expect. Next slide, please. Okay, so the second question is, what have been our biggest challenges? Well, probably at the top of the list is equipment failures. This is a recurring one. The intake tubing to the auto sampler gets clogged, or the tubing washes away altogether because of high flows in the manhole overnight. The battery that's been reliable for your peristaltic pump unexpectedly dies. The pH meter drifts, the 4-gas meter gives you a false positive, and so on. And so we do our best to plan better for the next time. We use a different strainer on the auto sampler tubing. We secure it with redundant hose clamps, and so on. Another challenge relates to safety considerations. You know, we're dealing with traffic while accessing manholes on city streets. We're dealing sometimes with very deep manholes and or manholes that have very high flows in them. We're dealing with thunderstorms, ice storms, very cold weather, very hot weather. And remember, we're dealing with sewage. And Steve talked a bit about this. Although not expected to be a route of transmission for COVID, sewage certainly has plenty of other transmissible pathogenic microorganisms in it. Another challenge are the sewage conditions themselves. So if the sewage is viscous, maybe because a lot of diapers are being flushed on site. If it's high in TDS, maybe because there's saltwater intrusion. Or if it's full of wipes due to the misleading idea that flushable wipes are flushable because they're really not. Any of these things can make sample collection difficult and also sample analysis difficult. And a final challenge relates to site access. You know, we might identify a manhole that we want to be sampling in, but that target manhole can be in the middle of a city street with 2 or more lanes of traffic, like is shown in this picture here. It can be hiding under a parked car, including under a police car in the middle of a bust. That's a true story. Or the manhole can be paved over. Also, that's happened multiple times. And then, of course, we have to go to a backup manhole. Next slide, please. All right, third, what innovations, if any, have we made? Now, I can think of a really big one here, and I have to warn you, it might not sound all that impressive to you, but let me assure you, it's been instrumental for implementation of our project in Michigan. Okay, so let's say you've decided you want to collect a wastewater sample from a particular manhole that captures just the neighborhood population you're interested in. The manhole is on this off to the side of a quiet road. It's not under a parked car, and it's not paved over. But it has one problem, and that problem is that the depth to the sewer in that manhole is 30 feet. Now, a typical peristaltic sample pump might have 20 to 25 feet of lift. But in this case, not only do you need to bring the sewage to the ground surface, you actually need to force it through a cartridge filter. Because let me remind you that right now, as of right now, there are many different ways to sample, prepare, and analyze wastewater for SARS-CoV-2 quantification. And it just turns out that the lab that will be performing the analysis on your wastewater sample prefers to have it filtered in the field to concentrate it. So you experiment with suspending different types of solids handling pumps down into the manhole to kind of act as a transfer pump. And you do it. You come up with this 2-stage pumping process. You actually fabricate not just the strainer on the end of that transfer pump tubing, but you sew a filter bag that fits right around the pump, because even though the pump is a solids handling pump, you're having problems with bird seed, of all things, mucking up your pump. But there you go, voila, you're in business. And so this is these pictures show this sampling system that we've been using in southeastern Michigan. So if anybody needs to filter samples from 30-foot deep manholes, come talk to us. We can help you set it up. All right, fourth question. What have we learned that we can take with us post-COVID? So one big picture lesson is that it's much better to engage with public health folks and officials sooner rather than later. If you aren't designing your sample collection program with input from public health people, then you may not be implementing a program that can effectively inform some of the public health interventions that Monina was talking about. Another kind of lesson learned, as I mentioned, there are things that are going to go wrong. Equipment will fail, manholes will be inaccessible, you'll have to deal with snow and hot weather and so on. So just have a backup plan. And finally, taking good field notes is critical. Sort of as the boots on the ground, when we see that the flow in a manhole is weirdly high, or there's a strange, a large amount of foam that's not usually there, or the sewage is this weird blue color. These are all things that are helpful to write down and report to the lab doing the analysis. All right, so fifth question, what are the limitations to wastewater surveillance, and particularly small scale surveillance? Well, it's messy. You're outside in the elements and in the snow and so on. It's murky. And what I mean by that is that it's not completely understood how best to use the wastewater data to inform public health interventions. And it can be slow. It takes 24 hours to collect a daily composite, if that's what you want to do, and then time to get that sample to the lab, and then time for the lab to get results to you. In an ideal world, you're getting results in no more than 2 days after sample collection, but we certainly live in a less than ideal world. Despite the messiness and the murkiness and the time-consuming nature of this, I've been told over and over by my colleagues that their participation in our wastewater surveillance work is the most meaningful work they've done as engineers. And that's all to say that it doesn't make it not worth doing, because wastewater surveillance certainly is worth doing. So next slide, please. I think what you've heard from our other panelists, and I'll reinforce this idea, is that this is really just the beginning of wastewater surveillance taking hold in the US. We know that HHS and CDC are committed to wastewater surveillance post-COVID, and we heard that from Monina as well. I think we'll continue to see surveillance at many scales, facility, city, regional, that incorporates both the PCR needed to quantify a specific target, but also the next generation sequencing that kind of looks and sees perhaps what other diseases may be circulating in a population, that looks not just at viruses, but also bacteria. And you've heard about antibiotic resistant bacteria. That's certainly of interest to the CDC, but also looks at substances. And Claire did a nice job covering the different substances, such as licit, illicit drugs that that wastewater surveillance can cover. And I could see a future where the US is divided up into large sewer sheds, and we're already kind of doing that with the HHSs with their 9-week program. But I'm showing another example here on the right. This is the Ain Ghazal sewer shed in Amman, Jordan, that has 2 million people living in it. And the idea would be that ongoing maintenance wastewater surveillance is done at this larger scale. But what if we took the effort before the next pandemic to identify the sub-sewer sheds that could be sampled to help pinpoint an outbreak? So that's what we did here in Amman. So we broke up this larger sewer shed into 4 zones, each of these 4 colors, and then we took each of those zones and divided it into 4 tiers, with the first tier having the most people in it, and then you get down to tier 4 where there's about 25,000 people corresponding to each sampling location, and each location is a manhole. So if we see something concerning at the larger sewer shed level, we would have a plan for how to quickly ramp up wastewater surveillance sampling in response. So with that, I'm going to turn it back over to Stephen. Thank you.
That was excellent. 4 different viewpoints on the state of the science, how we're using this, and where we might go for the future. So if you haven't already, use that question and answer function at the top or the bottom of your screen to type a question or a comment into the system. And Amy Miller, professor at Northeastern University, is going to be moderating question and answers. And we hope to get to all your questions. So jump right in.
Yeah, so while I wait for kind of more things to flow in, I think that there's a great question here to start us off. And this is probably for anybody on the panel who wants to jump in. And so this question is kind of thinking about if we want to implement this at scale to prepare for the next pandemic. And Anna, I think you just touched on that really nicely in terms of, given our current infrastructure, what might we do? But if the panel could think about, are there any suggestions you would have for how we might update our infrastructure, if there would be ways that we could kind of hone the system to have this be a built-in capability? And or are there certain skills that we need to be honing in our civil and environmental engineers to help this be a capability that we as a discipline can kind of roll out at the big scale? Yes, jump in. Go, Monina.
One of the things that I would say from the public health perspective is proper maps of coverage areas. Because when you need strategically to know where it is that you're covering, if maps were already available, that would put us ahead of time.
Yeah, that one's actually been discussed on a lot of different forums. Many utilities know where their pipes are, and they kind of know who they are sampling, but it doesn't necessarily match up with zip codes or Department of Public Health designations, or who's the local public health. There's a fair amount of overlap, and that is one of the difficulties in analyzing the data.
From the external kind of consulting and data science sides, are there any suggestions for sort of where the dividing line is of skills that are specialized and should be something that's considered outside of civil and environmental engineering, or things that are close enough to traditional disciplines that could be crossover skills?
Maybe I'm biased because I'm trained more from the bioengineering perspective, but I do think, reflecting on Biobot's experience, the folks on our team who aren't trained in a background in biology have had to learn it very quickly, and kind of, so some exposure to those, the laboratory assays that develop and generate the data, I think is useful, because it helps kind of, if you have an understanding sort of the process that generates that data, you can kind of understand, like, why is it so variable, or what type of variability do I expect, and what can it tell me and what can't it tell me? So I think having some exposure to those biological and chemical concepts is very useful. And I'll +1 the maps, and I think a component which I'm sure Anna has a lot of opinions on is just the basic like GIS knowledge, to analyze the flow, understand the catchment populations, and all of that.
All right, so an add on to that, I think almost every one of the speakers has mentioned something about potential for using this in the future. So could this be something that helps us identify or anticipate outbreaks in the future, or for other analytes, things like that? Are there, so I guess this is a two-part question. Monina, you specifically mentioned that these signals may precede the actual public health outcomes. So I'm curious if you might jump in and talk about what we would need to do to answer that question and use this as an anticipatory response. And then for the rest of the panel, I'm just curious if there are any technological gaps that you see as the major things we need to overcome before we could really roll this out on a bigger scale.
I'll start with the may part, and that is because unless the organism, or for example, in the case of the variants, the mutation is described, then it can't be detected in the wastewater. So it's not so much the place for discovering something new, but rather we would have to wait for the appearance of some clinical infections, identify the organism responsible, make sure that we understand the genomics around that, and then design assays to detect in wastewater. So that's where I was coming from.
And maybe just touching on this question of wastewater as a leading indicator of, disease resurgence or disease increases. This is actually something that Biobot has been asked kind of left and right. And where we've landed is that there's kind of different regimes and applications for wastewater epidemiology. And The one where the predictiveness is most useful and easiest to see is on a background of little disease activity. So I think, you know, kind of in the throes of the pandemic, when testing was really good, wastewater was really like a confirmatory signal of what the clinical data was showing. But as we're getting to kind of lower levels, or if we're sampling at a small facility like a building level where the baseline level is very low, that's when really you probably expect to see wastewater kind of pop up before a case, especially in a disease like COVID, where we know you start shedding before you start showing symptoms. So that means that you're starting to shed the virus before you're starting to, you know, you're seeking a clinical test. And so kind of in those situations where you have asymptomatic or pre-symptomatic shedding of whatever the causative pathogen is, I think that's where wastewater is really powerful. And the example Monina gave of polio virus, that's exactly kind of why polio is the first golden child of wastewater surveillance, because the majority of polio cases are asymptomatic. And so you wouldn't see them from people going to get tested, but you do see them in wastewater before there's an outbreak that happens. And so that's like really, I think, looking ahead to, you know, post-COVID, where it'll still be relevant for COVID and for these other sorts of diseases.
Yeah, I'll just add to that, Claire, that, you know, Monina touched on this as well, that I think wastewater is also mostly helpful when what's going on with clinical testing or diagnostic testing, if that's not as available as we would like it to be. So polio is a really great example. There's not a lot of it left in the world. There's not regular testing done for it. As you point out, they're asymptomatic cases. So you're right, that's a really great application of this tool.
So one follow on for that is, I think this was mentioned several times also, is just the nature of using PCR and using these sequencing techniques kind of builds in delays to data getting from place to place. And so I'm curious if any of you have thought about what would be potential technological pathways to shortening that. You know, is this just, you know, we're limited by the nature of PCR, or is most of that delay in the logistical challenges of sampling? Is there any kind of innovation if folks are looking for ways that they can contribute on research or engineering that we could potentially push forward?
Okay, I know I just answered the last question, but I'll start with an answer to this one as well, because you're right. I think a lot of the delays are in the logistics. You know, for our VA project, we have to send samples through FedEx. And, you know, I can tell you that the overnight guarantee is really not a guarantee anymore with FedEx. So one thing that the CDC is focused on right now is equipping wastewater laboratories with, you know, PCR capability if they don't already have it. And that Lumen Ultra gene count that I mentioned is one such device that's really well suited. It's, you know, it's small, it's relatively portable, but really well suited for use in an existing wastewater laboratory. So in that case, if a utility laboratory has in-house capability to do that PCR, and Hawk actually, you know, sells the Lumen Ultra device and packages up all the reagents you need to do SARS-CoV-2 analysis, I'm not sure I would call it quantification, you could be collecting the sample, running it, and getting the results the same day. So.
I will jump in a little bit as well. There are people who are working on alternate techniques that could be faster. You know, Aaron Bivens at Notre Dame has been working on protein-based as opposed to you know, genetic-based assays that lend themselves to something like an enzyme-linked assay that you know, people can run right in the field. It works just like, you know, your home pregnancy test sort of thing. And I believe also one of Biobot's neighbors in Cambridge has also been working on something like that. All
right, this one might be kind of a loaded question, but how much do folks see, or where do you see the balance of sort of the cost benefit of this all? Because I think our economy in general and our society in general really felt a heavy cost from SARS-CoV-2. And so cost was maybe not as much of a distinguishing concern this year when we really just needed solutions. But for something to be rolled out for public health monitoring in general, just as a preventative measure, there is definitely a question of how much that cost is. So I'm curious if folks have a sense of balance on where is the right kind of place that falls, and any suggestions for maybe pushing it to a cheaper direction. And Monina, it sounds like you want to jump in and start. Go.
Yes, so I showed that this is the one question that we have research going on in our shop, because we have to know whether it's cost effective for public health. Public health dollars are scarce, and if we don't use them wisely, and I think the answer is going to depend on the use case. So it could be that places where sampling is easier, for example, at the level of a wastewater treatment facility that doesn't require the gymnastics and all of that from a manhole, that might be something that is more cost effective, especially if it's in a location for like a Department of Corrections facility where clinical testing might be challenging, that might prove to be cost effective. But it could be that in another place, for example, the soldiers' homes, who have been screened extensively with clinical testing, and it's a smaller group, that maybe that's not going to be cost effective. But we have to answer that question. That's a very good question.
And maybe I'll jump in and say, I think one of the, now with kind of the high throughput biological assays and chemical assays, as long as you can get one wastewater sample, you can presumably measure kind of a very, very large number of things. And so I think like if we can get to a point where we're regularly generating all of this data, the cost effectiveness per data point will go lower and lower the more things you get out of each individual sewage sample.
I wonder if Claire and Monina might talk a little bit about some of the difficulties or around communicating the results, meaning what's the best question you've gotten from a reporter, perhaps, on the wastewater sampling? Go ahead, Monina.
I think this is where potentially we have to understand what the best strategy for communication is. So we haven't actually spoken to reporters. We instead just say, this is not something we're talking about, or refer to somebody else. And so we have potentially missed an opportunity to teach people how to interpret that information so that then they can go back and implement any further or reinforce some of the mitigation strategies that they're already doing. And I don't know if there are suggestions. I admire the efforts that MWRA has shown us as a model, that immediately results are available, they are shared publicly. So maybe what are people asking? Are they interpreting correctly? Yeah, I don't know the answer to that.
Claire?
Yeah, so, reporters don't usually, the reporters' questions get filtered and most of them don't make it to me. I think the hard ones are tackled by kind of our more seasoned folks. But, and before I answer that, maybe I'll add, the previous question about what's an important skill for civil and environmental engineers. I think for all engineers and scientists, communication is key, and learning to communicate to different types of audiences is really important. And without it, like, none of this will be successful. And I think, like, for me, the hardest question we get, and it's kind of an existential question, is like, well, what does this mean? Like, tell me this data point. What do I do with this data point? And the reason, there's like many reasons you can't answer that question. One is uncertainty in the data, variability in the data, just like all of the complex biology and engineering that's happening. And the other is kind of all the complex social and public health factors happening, where no data point can be interpreted by itself. And you know, we really view wastewater as one piece of the public health puzzle, but knowing how to act and what to do in response to it, like, it's not just wastewater that's going to tell you that. It's economics, it's behavioral science, it's kind of all these other things. And so when folks are really pushing us to tell, you know, like, tell me what this one data point means, I think that that's the hardest question to answer, because, you know, there really isn't one simple answer, but I think that's what people really want. So finding that balance is really tricky.
If I can add one more thing, my concern has been that where we leave an information vacuum, it'll be filled with misinformation. And so this is, I think, something that we need to work on.
Can I ask Steve and Steve a question? So I think I'll just echo what Monina said, that, you know, by you putting your data, MWRA, putting your data up on your website from very early on, I suspect you've been surprised by how different people have used it. So for example, the City of Cambridge Public Schools, I know they were looking at your trends in your wastewater as one of the 3 metrics to decide whether to open schools or not. Do you have other examples you could share of how sort of your data has been used that you weren't anticipating, either good or bad?
We definitely have a person who has contacted us at least a half dozen times asking us if we could format the raw data differently, which I suspect he wanted to be able to just, you know, automatically transfer our data to some other dashboard. And and to be honest, I don't know where he had intended to use it or how. We get questions about, you know, oh, the latest point went up, is that a trend? We have certainly had those questions from reporters. We get some highly technical questions that we tend to refer back to Biobot's public affairs people, and you know, and there's a lot of attention. If you follow our MWRA Twitter account, you will see that people frequently comment on the most recent data posts in a lot of different ways.
It's been one of the more popular items on our website over the last year, not surprisingly. And it was an explicit decision early on that if we were collecting the data, that the data had to be publicly available. And we worked out a strategy with the command center to make sure that somebody who might get a question had a few hours of lead time. We never want to be in a situation where someone at the Department of Public Health or the governor's office would receive a question about today's data before they saw the data. So we've been very careful about that. But we do think that public money is being spent on it, and it's public health, so it ought to be public. And it's been stuck up at the top of our website for how many, 15, 16 months now, Steve, something like that.
Yeah, not quite. June, so it's been a year.
Okay. It is the most popular. It is our most popular page.
That's actually a great transition. I had one more kind of note down here to follow up on. Steve put up a great map that showed all of the different locations that have been collecting similar data that provides some information online. Monina brought up the fact that there's similar monitoring in wastewater epidemiology for polio. I'm wondering if, just as a panel, you might have some suggestions for folks who are either interested in this specifically for SARS-CoV-2 or just wastewater epidemiology generally, if there are other resources you would point them to learn more, or what might folks might use as resources to familiarize themselves or maybe move their careers in this direction. Monina, I'll let you take it.
It might be a place to start, but CDC has a lot of actual training tools and things, webinars, literature with links to the actual papers. So that might be a good place to start. And that slide that I showed at the end where they're creating the National Wastewater Surveillance Network is intended to be a resource where people will be able to then observe data from other states and be able to do some comparisons and use that network as a resource for not only technical support, but maybe other advice and for information of context.
Yeah, CDC has actually put up on that website a number of nuts and bolts guidance documents. You know, this is how you collect samples. This is how you design your sampling program, or could. They're guidance documents, so you don't have to follow them exactly. These are the type of analyses that you either want to set up or work with a lab to get done. And here's some ways that you can use the data on the back end in terms of determining whether there's a trend and other approaches to use the analytics.
I might also mention that there's some good stuff on the Water Research Foundation's website looking at the sort of international experience on this. They've been instrumental in trying to take the state of the art and gather it together and then spread it out so everybody sees what's going on.
Stephen, I think I've gotten to the bottom of my list of questions, and I know we're approaching 1:30. I don't know if you had any other notes of things you wanted to toss to our panel for discussion.
Well, maybe at the risk of putting folks on the spot, has it lived up to its potential? Are we helping? Is all of this data helping us do a better job at managing the pandemic? Go ahead, Monina.
I would argue that it has, that it's been another point. The problem in epidemiology always is, if you only have one source of information, it can make you believe one specific picture. And having multiple sources of information that either follow a trend or something along the lines of what wastewater has done around cases, which is even potentially anticipating the appearance of cases, that this reassures or gives more confidence in any one of those sources, so that where there's a consistent pattern or places where they can supplement each other, wastewater has actually, I think, paid off well.
And I think from an implementation perspective, what's been so exciting to see is just the nationwide mobilization behind this technology. Like I mentioned before COVID, very, very few communities were doing this. And then with COVID, you saw kind of local municipalities take the lead in implementing this, either through folks like Biobot or their local university partners. Then you started to see kind of states catch up and start rolling out state level surveillance programs. And now, we've made it federal, and there's really robust systems in HHS and CDC, which, the CDC intends to continue funding. So just to see this technology go from like barely used to an actual part of the federal public health surveillance system, I think has just been remarkable and really shows that even if we haven't yet figured out exactly how to use it, I think that everybody's in agreement that there's huge value to the data and worth kind of investing in that infrastructure and that continued development of the technology.
If you don't mind, Steve, I might add leave us with a thought experiment. What if we had the wastewater surveillance infrastructure that we have now up and running last March? To what extent would that have been made it possible for us to avoid, shutdown and stringent measures in certain part of the country where we couldn't be certain that there was no disease, but that there was much lower incidence of disease than, say, in the Northeast, right? And then maybe we could have avoided sort of the COVID fatigue that occurred later in the pandemic. So.
It certainly perhaps could have been ramped up faster than clinical testing was, and had alleviated some of the pitfalls of the bias in that. So interesting. Well,
so I alluded to this in my talk yesterday. The NIH and a group at Johns Hopkins actually put out a press release and posted a paper indicating that the COVID virus may have been present in Boston, as early as, before Christmas of 2019. And so, we may have had to go back further than March to really make a big impact. But I think the difficulty in bringing up clinical testing and ramping up clinical testing to the levels that they are now, if we had the wastewater testing in place as a technique, could have helped to people to understand what the real dynamic was a lot better.
Mhm. Karen, do you want to send us off with a thank you to the panelists?
Yes, I want to give a very warm thank you to each of our speakers for participating today, and to Stephen Estes-Margiassi for leading our event, and Amy Mueller for leading the discussion. If you'd like to review a recording of the event or learn more about BSCES membership, please visit the BSCES website at bsces.org. This concludes today's presentation.